All right, hello there. This is Rachel Lytle from Brentwood, Tennessee, near Nashville, here to talk to you about meiosis and topic 5.1. So let's get started. What will we learn in this video? We'll learn the difference between diploid and haploid, the purpose of meiosis, the steps that make up meiosis, and what similarities and differences there are between mitosis and meiosis. So meiosis ensures the formation of haploid gamete cells in sexually reproducing diploid organisms. So we need to know what diploid and haploid are. So diploid means that there's two full sets or pairs of chromosomes in the cell. And those chromosome pairs can differ in size, shape, genetic information, and their centromere location. And this diploid cell contains two sets because it has one set from each parent. And it's represented by the symbol 2N. Examples of diploid cells are body cells, like skin cells in you, or leaf cells in a plant, or hypha cells in a fungus. Alternatively, haploid cells contain one set of chromosomes, and they're represented by the letter N. Examples of those are gametes or sex cells, like eggs or pollen. And the goal of meiosis is to make these haploid cells. So two haploid gamete cells can come together in sexual reproduction to produce a diploid cell. Meiosis results in daughter cells with half the number of chromosomes as the parent cell. So the diploid parent is producing 4 haploid daughter cells or sex cells. So if we look at this picture, we can see that we have a parent cell starting with a diploid number of 6, right? 2n equals 6. And you can count those in the image and see that there's 6 chromosomes there. And there's 3 pairs of them. And so after meiosis occurs, we should get these forming daughter cells. And notice there's 4 daughter cells that are forming here. And in each of those forming nuclei, we can see that there's 3 single chromosomes. And so these cells would be haploid with n equals 3. So in meiosis, there's going to be 2 rounds of division. The first of those is meiosis 1, and the first step of that is prophase 1. In prophase 1, we have the nuclear envelope beginning to disappear, fibers are forming, the DNA is coiling into these visible duplicated or double chromosomes that are made up of sister chromatids. Remember, the DNA is duplicated here because the cell has already gone through interphase and done DNA replication. And then we have the double chromosomes pairing up based on size, shape, centromere location, and their genetic information. And while they're paired up, the chromatids exchange genetic information with chromatids from the other chromosome. So you can see here that non-sister chromatids are what's exchanging genetic information, and they'll kind of swap segments. After that, we have metaphase 1, where the double chromosomes remain in pairs, and the fibers align them along the center of the cell. So you can see here they're in pairs in metaphase 1 across the center. After that, in anaphase 1, fibers are going to separate those chromosome pairs, and so we have these double chromosomes that are moving to opposite ends of the cell. In telophase 1, the nuclear envelope begins to reappear and establishes 2 separate nuclei. And each nucleus, you'll notice, has 1 double chromosome from those pairs. And so now at this point, at this point in meiosis 1 of telophase 1, the nucleus that's forming is only going to contain half of the total information that the parent nucleus contained. And so you can see here that 1 of each pair of double chromosomes is in the nucleus. So we started with 6, and now in each of these nuclei there will be 3. But notice that there's still double chromosomes, and that's why we need the second round of meiosis to occur, because even though we have successfully made daughter cells that have only 3 chromosomes in them instead of 6, they're still double chromosomes. So those chromosomes will begin to uncoil, cytokinesis will occur, and separate the cell into 2 daughter cells. And those daughter cells are, at this point, haploid and genetically different from each other and the parent cell. In meiosis 2, the first phase is prophase 2. You can see both of our daughter cells starting in that phase here. The nuclear envelope begins to disappear, fibers begin to form, and then we move on to metaphase 2, where now those fibers align the double chromosomes across the center of the cell. Notice those double chromosomes are now lining up single file when we're in metaphase 2. In anaphase 2, the fibers are separating sister chromatids. And so now in this phase, the chromatids or single chromosomes are going to migrate to opposite sides of the cell. In telophase 2, the nuclear envelope begins to reappear and establish separate nuclei. Each nucleus contains single chromosomes, and the chromosomes will begin to uncoil. Cytokinesis will separate the two cells into 4 daughter cells, and those daughter cells are now haploid and genetically different from each other in the parent cell. So you can see that we have 3 single chromosomes now forming in all of these new nuclei. All right, so here you see an image that has all of those phases put together. I'd encourage you to pause the video here and try to talk yourself through what we just went over before we move on. All right, so now we can compare mitosis and meiosis, and they're similar in the way that genetic information is passed on to the daughter cells. Both processes involve the nucleus disappearing, DNA coiling into chromosomes, aligning those chromosomes across the middle, and then using fibers to separate them, the nuclear envelope reappearing, those chromosomes uncoiling, and then cytokinesis producing the daughter cells. However, mitosis and meiosis differ in the number of resulting cells and the genetic content of those cells. So in mitosis, the goal is to make 2 daughter cells that are genetically identical to the parent cell. But in meiosis, we're producing 4 haploid daughter cells, and those are genetically different from each other in the parent cell. So let's practice. This video skill focuses on explaining biological concepts and/or processes. So here we have a practice question. Go ahead and pause the video and give it a try. And when you're ready, hit play and we'll go over the answer. All right, so the question asked which of the following best explains how meiosis results in 4 haploid daughter cells, and the correct answer is B. Meiosis involves 2 rounds of division, and double chromosomes remain in pairs during metaphase 1. The fact that they remain in their pairs during metaphase 1 means that they're going to separate those pairs, and that's when we get that haploid cells that result at the end of meiosis 1. And that's why B is the correct answer, because it is explaining how we're getting those haploid daughter cells. All right, so what should we take away? All right, key takeaways are that diploid cells have pairs of chromosomes and are represented by 2N, whereas haploid cells have a single set of those chromosomes represented by N. The purpose of meiosis is to produce haploid gametes. Meiosis has 2 rounds of division. In meiosis 1, we're separating the pairs of chromosomes, and in meiosis 2, we're separating the double chromosomes to get single ones. And mitosis and meiosis are similar in the way that they separate their genetic material and pass it on to the daughter cells. However, mitosis produces 2 genetically identical cells, while meiosis produces 4 haploid genetically varied ones. Right, and that's it for AP Daily today, and I'll see you next time. All right, hello there. Welcome to AP Daily. This is Rachel Lytle from Brentwood High School near Nashville, Tennessee, here to talk to you about topic 5.2, meiosis and genetic diversity. So let's get started. What will we learn in this video? We'll learn about crossing over, the random assortment of chromosomes in meiosis, and fertilization, and how all of those help to increase genetic diversity. So, meiosis results in 4 haploid gametes, or sex cells, that are genetically different. And there's these different processes that take place during and after meiosis that help to generate that genetic diversity. One of those is crossing over. So crossing over increases genetic diversity among gametes. And crossing over occurs during prophase 1 of meiosis 1. So in prophase 1, you can see in our figure that there are these double homologous chromosomes that are in pairs. And homologous chromosomes are chromosomes that carry information for the same genes, and you have one from each parent. So we've represented those here with different colors. So non-sister chromatids of those double homologous chromosomes actually exchange segments. And so you can see here in figure 2, where the red circles are, that the pieces of non-sister chromatids have broken off and switched places on those homologous chromosomes. And so what this results in is recombinant chromatids, meaning that they have a recombination of the genetic information that was compared to what was there before. And now with this formation of recombinant chromatids, we've increased the genetic diversity in these in these cells that will result from this process. All right, random assortment of chromosomes also is a way to increase genetic variation. So when the homologous pairs line up during metaphase 1 of meiosis, they can line up in different orders. So here we see a possible way that the chromosomes that we were just looking at and their homologous pairs could line up during metaphase 1 of meiosis 1. Here we see another way that they could possibly line up. And so you'll notice, looking at these red circles, that the one on the left had all of the yellow chromosomes on the left and all of the blue ones on the right, versus in the second image on the right, we can see that the two of them have swapped places. And either of these are possibilities as to how these homologous pairs could line up during metaphase 1. You can see crossing overs continuing during metaphase 1. And as we go through the stages after metaphase 1, we'll see how the way that they line up could result with different combinations of chromosomes in the resulting gametes. So if we go from metaphase 1 here to what anaphase 1 would look like for both of those cells, we see those homologous pairs have separated. And then if we go all the way to the end result of what the gametes would look like and what chromosomes would end up in each gamete based on the order that they lined up in earlier, we can see that there are differences, right? So the gamete on the left and the gamete on the right have different chromosomes in them, and that's just based off of the fact that those homologous pairs lined up in a different order during metaphase 1. And if we go through and compare all the resulting gametes that would be produced during this round of meiosis, we'll see that they differ in each one of those cells. Right? Fertilization of gametes can also serve to increase variation. And so when fertilization occurs, information from each parent is contributed to the fertilized egg. And typically you have one gamete from each parent that fuse together and those form a diploid offspring. Fertilization is random in that any gamete can contribute to the diploid nature of genomes in the offspring, and this increases the potential for genetic diversity. So if we look back at our model and we see figure 1, we have the gametes that we produced earlier, and we'll say that those are the gametes from parent 1. In figure 2, we can see gametes that were produced by a different parent. And so if there was going to be an offspring produced from parent 1 and parent 2, any one of these gametes could be used to produce that offspring. So let's say that we have these two gametes that come together, and this is our potential offspring that is produced. And you can see by all the different colors and the results of crossing over that there's a lot of genetic variation within that offspring. So let's practice. Right, in this video, our lesson skill focus is questions and methods. So we want to identify or pose a testable question based on an observation, data, or a model. So here we have our practice question. It's showing us figure 1, and it's asking you which one of these questions could most appropriately be answered by looking at the model in figure 1. So go ahead and pause the video here and give it a try, and then we'll come back together and talk about it. All right, so the correct answer to this question is choice C. How does the independent assortment of the two sets of homologous chromosomes increase genetic diversity? And the figure is giving us a diagram of that random assortment of chromosomes and two different ways they could have lined up during metaphase 1, and the resulting differences in the chromosomes that end up in the gametes at the end of the process. And so we know that that's the correct answer because it could be answered from this figure. Some of the other questions mention processes that aren't shown in the figure. For example, choice A says crossing over, B says DNA methylation, and D talks about zygotes, which are produced during fertilization. So those three could not be answered by this figure. Right, so what should we take away? Key takeaways are that crossing over occurs in prophase 1, and that's when non-sister chromatids exchange segments, and that results in recombinant chromosomes, and that recombining of the chromosomes gives us genetic variation. Random assortment of chromosomes in metaphase 1 can result in different combinations of chromosomes that end up in those gametes. And during sexual reproduction, any gamete from one parent can combine with any gamete from another parent, and that results in genetically different offspring. This increases the genetic diversity within a population of organisms. Right, and that's all we have for AP Daily today. I'll see you next time.
All right, welcome everybody to AP Biology. This video will explain how chromosomal analysis can show patterns of inheritance. The skill we're using for this video is predicting the effects of disruptions to biological systems using visual representations. So let's get to it. What will we learn? In this video, we will learn three important things. How can chromosomal inheritance show patterns of transmission of genes from parents to offspring? How can human genetic disorders be based on chromosomal inheritance? And how can we use visual representations to analyze changes or disruptions in chromosomal inheritance? So let's get to it. The chromosomal basis of inheritance provides an understanding of gene transmission. And what I mean by this is that certain genetic disorders can be caused by a single mutated allele or a specific chromosomal change that is passed from parents to offspring. This parent to offspring inheritance can be analyzed to determine the pattern of that gene expression, of gene transmission, And mutations or misformations in gametes can result in disorders being present in offspring that were not present in parents. So let's look at an example of this. Huntington's disease is a disease you might have heard of, you might not have. It's a progressive and eventually fatal neurological disorder. This is caused by a single defective gene on chromosome number 4. And the inheritance pattern here is autosomal dominant, meaning if you inherit the affected chromosome from a parent, you will get the disease. And we can analyze this inheritance by looking at a figure called a pedigree, which is what's shown here. And if someone is affected with Huntington's disease, their symbol is shown as colored in here. And what we can see if we look at the oldest generation, we had 3 out of their 5 children ended up having that disorder. Given that it's a dominantly inherited disorder, that makes sense. And then following that down, those individuals who were affected tended to have affected children. 2 non-affected parents, like you can see right there in the middle, had non-affected children. So let's practice this. Let's give you guys a situation and see where your thinking is. The skill that we're going to be focusing on with this practice is argumentation. That means the question is going to ask us to predict, bold word is predict, the causes or effects of a change in or disruption to one or more components in a biological system based on a visual representation of a biological concept, process, or model. So here's a question for you. We have a visual representation right there in the middle, information up above, and the question with that bold word predict down below. So pause the video, read through all of this information, come up with what you think is the best answer, then unpause and we'll talk about it. All right, welcome back. Hopefully you have read through this situation involving hemophilia. The prompt tells us a lot of information that's necessary for us. So let's read that. It says that sex chromosomes determine the phenotype of sex in humans. Embryos containing XX chromosomes develop into females, and embryos containing XY chromosomes develop into males. The sex chromosomes separate during meiosis, going to different gamete cells. And that's what they're showing us in this picture. They are showing us this woman, XX, and her 2 gametes that are formed. A woman is heterozygous for the X-linked recessive trait of hemophilia A. Her sex chromosomes can be designated X capital H, X lowercase h. During meiosis, the chromosomes separate as shown in figure 1. So that's what we're seeing here. We are seeing this X capital H, X lowercase h heterozygous woman making 2 gametes, one containing each of her X chromosomes. If the woman and a man with normal clotting function have one biological daughter and one biological son, which children are predicted, remember we're being asked to make a prediction, to have hemophilia A? So my hope is that the son was your answer. The son is the only one that could end up with this recessive and hemophilia chromosome. His other chromosome would be the Y coming from dad, which is not able to give him a capital normal clotting allele there. So our big takeaways from this video are that chromosomes are inherited as full units and passed from parents to offspring. The pattern of inheritance of traits, diseases, or disorders can be analyzed to determine how these chromosomes were inherited. And visual representations such as pedigrees can be used to help us predict the causes or effects of changes in chromosomal inheritance from parents to offspring. Thank you very much for spending the time with me to watch this video. If you still have questions, feel free to go back, rewatch the video again, take some notes. Good luck. Welcome everybody to AP Biology. My name is Lisa Niesman. I'm coming to you from Bay Ridge Preparatory School in Brooklyn, New York. This video will explain how inheritance of chromosomes results in genetic variation. The skill we're going to be targeting is predicting the causes of disruptions to biological systems using visual representations. So let's get started. What will we learn in this video? We're going to look at 5 different things. The first is, how does the law of segregation account for genetic variation? The second, how does independent assortment result in genetic variation? What is non-disjunction and how does it contribute to genetic variation? I hope you're starting to see a theme here. Number 4, how does fertilization in sexually reproducing organisms lead to genetic variation? And finally, what effect does chromosomal inheritance have on genetic variation and human disorders? The theme, as you can see, is chromosomes and genetic variation. So let's get to it. There's a lot of text on this slide, and I promise we're going to go through it. So the first 3 pieces of this that we're going to talk about are segregation, independent assortment, and random fertilization. So the law of segregation explains the separation of alleles during gamete formation. What this means is that each gamete carries only one allele for each gene. Therefore, each gamete receives only one parental allele. This segregation of parental alleles into gametes provides an opportunity for more varied combinations of alleles when fertilization occurs. Take a breath. The second piece of this is independent assortment. So what independent assortment does is it suggests that genes for 2 or more traits will be sorted into gametes independently. This means that the genes are not linked, they're not inherited together. Inheritance of each gene, therefore, is random, and it's not connected to the inheritance of any other gene. This assortment of genes independently into gametes provides more possible gene combinations when fertilization occurs. An example to help you guys think about this is pea color and pea shape in Mendel's famous experiments. The color doesn't determine the shape. Those are independent genes. Random fertilization refers to the concept that any of the genetically unique sperm created by a male can join with any of the genetically unique eggs created by a female. This is going to produce offspring with a genetically unique combination of chromosomes. A lot of text on this page, but what we're focusing on is these different mechanisms that can result in genetic variation. So we oftentimes look at this with genetic disorders, many human genetic disorders can be attributed to the inheritance of a mutated allele. And what we mean by this is that the chromosomes can carry these mutated alleles. The laws we just talked about, segregation and independent assortment, those are going to explain how these mutated alleles are randomly distributed into various gametes. And these mutations then can contribute to genetic diversity in the offspring. These mutations can manifest as genetic disorders if the variations negatively affect the functioning of offspring. So there's one more piece of this. These disorders can be attributed to specific chromosomal changes. Non-disjunction is the term we use for a failure of chromosomes to fully separate during the formation of gametes. And what this means is the sex cells are going to end up with either too few or too many chromosomes. And that's what this picture here is representing. It's representing N being a traditional number, in this case 1, whereas here you're seeing non-disjunction. This chromosome and this gamete is going to end up with more than the expected number of chromosomes. So let's look at an example of this. Examples always help us clarify confusing content. So an example is something called triple X syndrome. So what happens with this genetic disorder is that there's a malformation either in the egg or sperm cells. This is an example of non-disjunction, resulting in the reproductive cells having more than one chromosome. What that means is the offspring is then going to have more than 2, or the standard number of sex chromosomes. So you can see here, this individual has 3 X chromosomes, hence the name triple X syndrome. So let's practice this. Let's look at a question and see how your thinking has been clarified about this content. The skill that we're going to be working on here is argumentation, which means that we're going to be predicting the cause or effect of a change in or disruption to one or more components in a biological system based on a visual representation of that biological concept, process, or model. So our bold word is to predict, and we have a visual representation coming our way. So here's that visual representation. Here's a question, and you'll notice that word prediction is in bold. So I'd like you to take a look at this question, pause the video, try to come up with what you think is the best answer. When you have it, unpause the video and come back to me. All right, I hope you have taken the time to analyze this beautiful karyotype picture showing different chromosomes and to read the question. So the question says, which of the following represents the most accurate prediction, remember that was our verb, of the cause of the inheritance pattern illustrated in this karyotype? My hope is that answer choice C is the one that you guys all jotted down. The chromosomes failed to separate during gamete formation. That's how this individual has ended up with X, X, and Y, 3 sex chromosomes. So our key takeaways, what are we taking away from this video? Remember, we had 5 of those what will we learn statements. Let's answer each one of them. The segregation of parental alleles into gametes provides opportunity for more varied combinations of alleles when fertilization occurs. Remember our goal of genetic variation here. Number 2, the assortment of genes independently into gametes provides more possible gene combinations when fertilization occurs. Again, independent assortment leading to this genetic variation. Number 3, non-disjunction is the failure of chromosomes to fully separate during the formation of gametes. This results in too many or too few chromosomes in our sex cells. Random fertilization leads to genetic variation in offspring, specifically of sexually reproducing organisms. And lastly, certain human genetic disorders can be attributed to chromosomal inheritance and can result in genetically varied offspring. I hope this video helped clarify a couple of concepts for you. If you still have questions, feel free to go back and review the video. Good luck. See you for the next time.
All right, hi there. Welcome to AP Daily. This is Rachel Lytle from Brentwood High School near Nashville, Tennessee, here to talk to you about topic 5.3, Mendelian genetics. So what will we learn in this video? We will learn how conserved processes support the concept of common ancestry and why those processes are considered fundamental for the continuity of life. So here we go. Heritable information provides for the continuity of life. So nucleic acids like DNA and RNA are carriers of genetic information. And you can see in this image, we have a model of those. And the nitrogenous bases that are parts of those molecules and the order of those nitrogenous bases carry genetic information. And that genetic information is transferred to other cells during cell division. And transmission of genetic information can be also from one generation to the next, and that helps to provide continuity of life. There are shared, conserved, fundamental processes and features that support the concept of common ancestry for all organisms. So we're going to look at a couple of these. So one of those major features is the genetic code, and that's shared by all modern living systems. All organisms use nucleic acids to store and transmit genetic information. You can see in this image that we have the nucleic acids, DNA and RNA, and they're involved in that transmission of the genetic information. All organisms also have ribosomes, and they use those ribosomes to synthesize proteins based on nucleic acid sequences. So let's look at an example. Here you see three images, one of a bacterial cell, an animal cell, and a fungal cell. And if you compare the three images, you should notice that all three of them contain ribosomes. And so that shared feature of containing ribosomes and using them to build proteins supports the concept of common ancestry for all organisms. Right, there are also some shared processes like core metabolic pathways that are conserved across all currently recognized domains of life. So if we look at an example of one of those, we could look at cellular respiration, which is a process used by all organisms. And here we've got images of anaerobic respiration and aerobic respiration, so two different types. But if you compare the two, you should notice that glycolysis is in both, and it's a conserved aspect of cellular respiration. So the fact that it's in both can support the concept of common ancestry for all organisms. All right, and now it's time to practice. So our lesson skill focus for this video is argumentation and specifically predicting. So we want to predict the causes or effects of a change in or disruption to one or more components in a biological system based on data. So here we have our practice question. Go ahead and take a minute to pause the video, answer it on your own, and when you're ready, press play. All right, so the question is asking us which of the following best predicts the effect of inserting this gene into the DNA of a bacterial cell. And the correct answer here is A. The recombinant bacterium will produce human insulin using its own machinery. So since the genetic code is one of those shared conserved processes across all organisms, if we insert DNA from a different organism into another one, it has all the machinery and the mechanisms necessary for it to create that protein. And so that's why A is the correct answer. All right, so what should we take away from this video? Our key takeaways are that there are shared and conserved processes that support the common ancestry concept because fundamental features link organisms by lines of descent. And processes like cellular respiration and the transmission of genetic information allow for life to continue through generations and give you that continuity of life. All right, that's all for AP Daily today, and I'll see you next time. All right, hello there. This is Rachel Lytle from Brentwood High School near Nashville, Tennessee, here to talk to you about topic 5.3, Mendelian genetics. So what will we learn in this video? We'll learn about Mendel's laws of segregation and independent assortment. We'll learn about monohybrid and dihybrid crosses, how the laws of probability can be applied to genetic crosses, and what a pedigree is and how the patterns can show types of inheritance. So first we'll look at Mendel's laws, which describe the inheritance of genes and traits on different chromosomes. A gene is a unit of heredity that codes for a trait, and it can be transferred from one generation to the next. A trait is genetically determined characteristic of an organism, and genes determine these traits. Right, an allele is a specific variation of a gene, and organisms inherit their alleles from both parents. Organisms can inherit different alleles for the same gene, and they're often represented using letters, like a capital letter and a lowercase letter. A dominant allele is what shows up in the phenotype if it's inherited and is represented by an uppercase letter. A recessive allele only shows in the phenotype when the dominant allele has not been inherited, or it's a lowercase letter. A genotype is the combination of the alleles that you inherit, and it's typically represented by two letters. If you are homozygous for your genotype, that means you contain two of the same alleles, so either maybe both dominant or both recessive. And if you're heterozygous for that genotype, then the genotype contains two different alleles, so maybe a dominant and a recessive allele together. The phenotype is the physical result or expression of your genotype. All right, so first we're going to look at the law of segregation. So we need to know that chromosomes carry alleles, and homologous chromosomes carry alleles for the same trait. So if I look at this image here, I have a pair of homologous chromosomes, and you can see that there are alleles inherited on those chromosomes from each parent. And when those chromosomes are separated into daughter cells during meiosis, the alleles for each trait are also separated so that we can see that homologous pair and the alleles that are on those chromosomes have been packaged into two different gametes from that parent. So if the same thing is happening with another parent, and then these gametes are going to combine for fertilization, we can see that there are a lot of different possible combinations of alleles that result because those alleles were separated originally in the parents during meiosis. And so the separation of alleles allows for genetic variation among the gametes and then eventually among the offspring. So let's look at an example of that. Mendel's experimental designs involved specific crosses of different generations. So in his parent generation, or the P generation, he had a homozygous dominant crossed with a homozygous recessive, and we can see that here in the first Punnett square. And in the F1 generation, all of those resulted in heterozygous purple flowers. And then in his F2 generation, he crossed two heterozygotes, and you can see that because we have this reappearance of a white flower, which is recessive in that cross, that's evidence of the law of segregation. That wouldn't have been possible if it wasn't for the fact that those recessive alleles from each parent were packaged into separate gametes from the dominant alleles. All right, Mendel's other law that we're going to talk about is the law of independent assortment. And for this one, we're going to talk about two or more genes and how they assort independently of each other. So here we have a hypothetical gene A and gene B. One trait does not have to be automatically inherited with another trait. And so these alleles for the separate traits can be packaged in every possible combination into the gametes. And we're going to want to give one allele for each trait, so one A and one B to each gamete. So with this parent that's heterozygous for both traits, there are a lot of possibilities. They could give it a dominant allele for A and B to the gamete. They could give a dominant A and a recessive B. They could give a recessive A and a dominant B, or they could give both recessive alleles. We can also use rules of probability to analyze the passage of traits from a parent to offspring. And so understanding Mendel's law of segregation allows for the use of mathematical calculations to determine the probable inheritance of an allele. So let's look at an example of that. If we're looking at probability and we want to determine the probability of event A or event B happening, we want to add the probability of event A to the probability of event B. But if we want to determine the probability of event A and event B happening, we're going to multiply those probabilities together. So we're looking for the difference between or. If you have or, that means that you're going to add. If you're talking about event A and event B, so the and, that means you're going to multiply. And this is also provided for you on your AP Biology formula sheet during the exam, and it looks like this on the formula sheet. So if we look at a question here, it says, assume that genes A and B are not linked. If the probability of allele A in a gamete is 1/2 and the probability of allele B in a gamete is 1/2, then what is the probability of both A and B being in the same gamete? And so since it says the probability of both A and B, and it said and, we're going to multiply those probabilities. So if we multiply 1/2 times 1/2, we would get our answer of the probability is 1/4. We can also look at different crosses. A monohybrid cross is an examination of how one trait is inherited, right? Mono means one. We can also have dihybrid crosses, and di means two. So a dihybrid cross is an examination of how two traits are inherited. Punnett squares are typically what we use to illustrate the probable outcomes of a cross. So let's look at an example of a monohybrid cross using Punnett squares. Right, in this cross, we're looking at one trait, and that trait is flower color. That one trait has two possible phenotypes, purple flowers, which are dominant, or white flowers, which are recessive, and there's 3 possible genotypes. So we can see that these Punnett squares are showing possible offspring of the parental cross, and those possible offspring are what are filled in inside the Punnett squares. And we can talk about those in terms of percentages as well. For this F2 generation, I could say that there's 3 purple to 1 white phenotype ratio, or I could say that there's a 25% chance of those parents producing a white flower offspring. Patterns of inheritance can often be predicted from data including pedigrees. And a pedigree is a visual representation that traces the history of a trait through familial generations, and it helps us identify the types of inheritance. So here is a pedigree, and you'll need to know that circles represent biological females and squares represent biological males. Shaded shapes mean the individuals are affected, and unshaded shapes are unaffected individuals, and that means affected by the trait. They have the trait that we're following in the pedigree. Perpendicular lines that branch off of horizontal lines represent offspring. So you can see here that there are 3 offspring, and those would be the offspring of those 2 parents at the top. Generations can be indicated with Roman numerals. So you can see in this pedigree, we have 3 different generations. And then if we want to talk about a particular individual, we can talk about them based on their number. So if I say individual 3-2 or generation 3, individual 2, I am talking about this individual male right here. So let's look at an example of a pedigree and how we could use that to determine the pattern of inheritance. If we have an autosomal dominant trait, autosomal meaning it's on a non-sex chromosome, then it's going to show a pattern of affected offspring that have affected parents. And if we look at autosomal recessive traits, we'll see that there's a pattern of affected offspring that have unaffected parents. So if we look at this pedigree, we notice that there are some children here, some offspring that are affected by the trait, but their parents are not. So that is a pattern that is indicative of autosomal recessive traits. So what should we take away? Our key takeaways from this video is that Mendel's law of segregation states that alleles can be segregated into separate gametes during meiosis. The law of independent assortment states that genes are not linked and they can be divided independently into different gametes. A monohybrid cross examines one trait's inheritance, while a dihybrid cross examines 2. Understanding Mendel's laws allows for the application of mathematical calculations and probability. And pedigrees show us the inheritance of patterns of families and can be used to predict inheritance traits in subsequent generations. Thanks for watching AP Daily, and I'll see you next time. Hi, this is Rachel Lytle from Brentwood High School near Nashville, Tennessee, here to talk to you about topic 5.3, Mendelian genetics, specifically about chi-square hypothesis testing. So what will we learn? Why do scientists use hypothesis testing? What is the purpose of a chi-square goodness of fit test, and what steps are necessary to perform it? All right, so hypothesis testing is used by scientists to reject or fail to reject statistical hypotheses, and it helps to determine if differences in numerical data are due to the independent variable being tested or if they're just due to chance. So there's two types of hypotheses. The null hypothesis states that there is no relationship or no difference between two groups of data in an investigation. So think null means no relationship. The alternative hypothesis is one of many possible hypotheses that states observed results are due to some non-random cause. So a chi-square goodness of fit test is going to be used to determine if there's a significant relationship between two groups of data. And to do that, we need to compare observed outcomes to expected outcomes and see if the variations in them could be due to chance. And it's often used in biology to test genetic crosses, but it can be used for other analyses as well. So the steps to perform a chi-square goodness of fit test are, number one, establish the research question, determine your null and alternative hypotheses, count the observed values, determine expected values, use those to calculate the chi-square value using this formula, calculate the degrees of freedom, select the p-value, identify the critical value from the table, and compare the chi-square value to the critical value to draw your conclusions. So we're going to look and go through an example. So our research question that we're going to ask is, do seed color and seed shape assort independently? To do that, we're going to look at dihybrid corn. Dihybrid, remember, meaning there's two traits involved. So the first is seed color, and the dominant trait for seed color is purple, which is because it's pigmented, or the recessive trait is yellow, which means it's lacking pigment. Our second trait is seed shape, which is dominant for smooth, which is because it's starchy, or wrinkled, which is recessive, and that's because it's sweet. So if we look at what those phenotypes look like on our dihybrid corn, you can see that there are different examples here: yellow smooth, purple wrinkled, purple smooth, and yellow wrinkled, and that's what they appear to look like. So next we need to determine our null hypothesis, and that's going to be that there's no relationship between seed color and seed shape, while our alternative hypothesis is that there is some sort of relationship between seed color and seed shape. So next you need to count the observed values. What did you actually observe in the corn? So I'm going to count the number of seeds that have each phenotype, and I counted 5 rows of corn kernels here. And I'm looking for these phenotypes, and when I counted them, this is what I observed: 118 purple smooth, 29 purple wrinkled, 33 yellow smooth, and 6 yellow wrinkled. So next I need to determine my expected values, and this is where most of my students have trouble. So we need to complete a dihybrid cross for the traits of interest to come up with what we would expect of these values if these traits do assort independently. So I'm doing a cross, and I know that the genetic corn that I have was a result of the cross between two parents that were heterozygous for both of these traits. And I'm going to make a dihybrid Punnett square to come up with my expected values. So I know that these parents with this genotype can assort their alleles into gametes and any possible combination, and these would be the possible combinations from this parent. And this parent has the same genotype, so I know that these are the possible combinations that that parent could give to an offspring. And so then I'm going to start filling in what the possible offspring could be depending on which gametes come together during fertilization. So I'm going to keep my R's together and my SU's together, and I'd encourage you to pause the video here and fill in the rest of the table by yourself. All right, but I'm going to go ahead and there we go. That's what it would look like. And then we're going to want to determine the phenotypes because that's what I counted for my observed values. So I'm going to determine the expected phenotypic ratio. And this one I know that purple was dominant and smooth is dominant. So since I have homozygous dominant here and homozygous dominant there, I know I'm going to have a purple smooth kernel or seed. And I can fill in the rest of them, and that's what I get as my expected results. So I would expect to have 9 purple smooth to 3 purple wrinkled to 3 yellow smooth to 1 yellow wrinkled. And so now I need to make those into values that I can compared to my observed values. So my observed values, if you total them up, added up to 186. And so for my expected values, well, out of 186, I would expect 9 out of 16 of those to be purple smooth. So I'm going to multiply 186 by 9/16, and I would get a number of 104.6. It's okay to have a 0.6 of a seed in an expected value because it's not something you actually observed. All right, for purple wrinkled, it would be 3 out of 16. Yellow smooth would also be 3 out of 16, and yellow wrinkled would be 1 out of 16, and so that's how I determine my expected values. I'm also going to double check myself and add those back up together and make sure that they total the same total as my observed to make sure I didn't make any mistakes there. All right, then I'm going to calculate the chi-square value. So I'm going to plug these observed and expected values into the chi-square formula. So here I've combined my table that has my observed and expected values I just came up with. And this chi-square equation is provided to you on your AP Biology formula sheet. And what we're going to do is the observed minus the expected squared over the expected for each of these different phenotypes, and I'm going to add them all up to get a number. So if I'm looking at purple smooth, my observed was 118. So my observed minus my expected squared over my expected. And then I do that for every group, right? All of these other different phenotypes and add them together to get my chi-square value, which is 5.52. Next, I want to calculate the degrees of freedom. This is also on your formula sheet. It says the degrees of freedom are equal to the number of distinct possible outcomes minus 1. I had 4 possible outcomes, right? 4 phenotypes minus 1, so my degrees of freedom is 3. I'm going to select a p-value. So notice in this table, also provided on your formula sheet, that there are 2 possible p-values, and I'm going to select a p-value of 0.05. I'm going to identify the critical value. So I'm using my table. If I had 3 degrees of freedom and a p-value of 0.05, my critical value is going to be 7.81. So in my last step, I want to compare that chi-square value to my critical value to draw conclusions. And this is something that you have to remember right here. If the chi-square value is less than the critical value, then you're going to fail to reject the null hypothesis. If the chi-square value is greater than the critical value, then you're going to reject the null hypothesis. So I had a chi-square value of 5.52, which is less than the critical value of 7.81. And that means I failed to reject the null hypothesis. So I failed to reject that there is no relationship between seed shape and seed color, which means I'm going to conclude that the 2 traits assort independently of each other. All right, now we have a practice question, and our skill is to perform chi-square hypothesis testing. So pause the video here, take a while to look at this question and do your calculations, and then press play when you're ready to see the answer. All right, the correct answer was A. The calculated chi-square value is 9.24 and the critical value is 7.82. So the null hypothesis of independent assortment can be rejected. All right, what should we take away from this video? Scientists use hypothesis testing to determine if results are due to the independent variable or due to chance. The chi-square test evaluates numerical data from 2 groups to determine if they are significantly varied from the expected results. And the steps of a chi-square test include establishing a question and hypotheses, determining the observed and expected values, calculating the chi-square value, identifying the critical value, and then drawing a conclusion as to whether the null hypothesis should be rejected or if the data will fail to reject the null. And that's it for AP Daily, and I'll see you next time. Hi there, welcome to AP Daily. This is Rachel Lytle from Brentwood High School near Nashville, Tennessee, here to talk to you about topic 5.4, non-Mendelian genetics. So what will we learn in this video? We will learn what linked genes are, what map distance tells you about a pair of linked genes, what sex-linked traits are, and how those sex-linked traits differ from Mendelian traits. Right, patterns of inheritance of many traits do not follow the ratios predicted by Mendel's laws. Genes that are adjacent or close to one another on the same chromosome and that are inherited together are known as linked genes. Traits that are determined by genes located on sex chromosomes are known as sex-linked traits. And these linked genes and sex-linked traits do not follow the predicted ratios associated with Mendel's laws, and they can be identified by quantitative analysis. Genes that are adjacent and close to one another on the same chromosome may appear genetically linked, and they're typically inherited together. That means they're less likely to be separated during crossing over in meiosis. So if we look at this model, we have a pair of homologous chromosomes, and you can see gene A and gene B there are very close together on the same chromosome. So even as these homologous chromosomes go through crossing over in meiosis, it's very unlikely that those genes, gene A and gene B, will be separated because they're so close together on the chromosome. So they're most likely to be inherited together. The probability that genetically linked genes will segregate as a unit can be used to calculate the map distance between them. So map distance tells you how close together a pair of linked genes is, and it's determined by how frequently a pair of genes participates in a single crossover event. And so linked genes have a recombination frequency of less than 50%. So let's look at an example. We have a pair of linked genes that has a recombination frequency of 5%, it's very low, then they are close together on the chromosome. It means they only separate 5% of the time. And these linked genes are considered 5 map units apart. If we look at a pair of linked genes that has a recombination frequency of 30%, that means they're further apart on the chromosome. And these linked genes are considered 30 map units apart. And you can see now in this figure that gene A and gene B are further apart, still on the same chromosome, but they're further apart. And so there is a little bit more of a chance that they could be separated. And so map units can be used to determine the distance between genes on a chromosome. Some traits are determined by genes on sex chromosomes, and those are known as sex-linked traits. Sex-linked traits are located on the sex chromosomes, and sex chromosomes determine the biological sex in animals. Sex chromosomes are non-homologous, and they have different letter designations. In humans, that's X and Y. Some other organisms use Z and W. Sex-linked traits, like we said, deviate from Mendel's model of inheritance. So the pattern of these sex-linked traits can be predicted from data, including pedigrees. And sex-linked trait pedigrees have their own particular pattern that you can identify. So let's look at an example of sex-linked traits, specifically in humans. In humans, biological females contain 2 X chromosomes, and biological males contain an X and a Y chromosome. The Y chromosome is smaller and carries very little genetic information. Therefore, most of the sex-linked alleles that we look at in humans are carried on the X chromosome. Alleles are shown as a superscript on the X chromosome. So if you look at this table here, you can see the possible genotypes of sex-linked traits in humans for some gene A. And you can see that there are 3 possibilities for biological females, and there's 2 possibilities for biological males. Also notice that the Y chromosome does not receive an allele because it does not carry this trait. Biological females can be considered carriers or heterozygous of a recessive sex-linked allele. Because biological females have 2 X chromosomes, they can be heterozygous. There's no way for a biological male to be heterozygous or a carrier because biological males only have 1 X chromosome. Biological males are more likely to have phenotypes associated with recessive sex-linked alleles for this reason, because biological males only have to inherit 1 copy of the recessive allele in order for it to show up in their phenotype. So let's look at an example. The pattern of inheritance of sex-linked traits can be predicted from pedigrees, as we said, and so this is the sex-linked recessive pedigree. You can see that it's recessive from the pattern of the affected offspring having unaffected parents, like we see here in the red circle. And you can notice that biological males are more likely to be affected than biological females, and in this example, only biological males are affected. Sometimes biological female carriers are shown as half-shaded circles, although not in this example, but you may see that sometimes. And some genotypes or phenotypes can be determined looking at the pattern of this pedigree. So I can identify that this biological male right here has a genotype of X with a recessive A and Y because he is a shaded in square, and so that means he is affected with the recessive trait. Could also identify that this female, or this biological female, his mother, has a genotype of a carrier or is heterozygous because she had to provide the recessive allele to her offspring or him. And so we know she has at least a recessive allele in her genotype. But We know that she's not shaded in, so she does not have the trait, so that means that she must be a carrier or be heterozygous. All right, so let's practice. Our skill focus for this video is to perform mathematical calculations, including means. So here is your practice question. Take a second to pause the video and read it over. Give it a try, and when you're ready, press play, and we'll talk about it. All right, so this question is asking which of the following is the mean number per cross of F2 generation offspring that are a result of crossing over. So if we had crossing over occurring, we would expect to see phenotypes that are different than the parental phenotypes. And so I'm looking at these two rows here. And to take the mean, I'm going to add them up and divide by the number of crosses. So I need to add up for cross 1, it would be 1. For cross 2, we have 3. For cross 3, there's 1. For cross 4, there's 3. And for cross 5, there's 3. And so if I add those all up and divide by 5, because there's 5 crosses, I get that the answer is B, 2.2. All right, what should we take away from this video? Our key takeaways are that linked genes are genes located on the same chromosome that are typically inherited together. Map distance tells you how close together a pair of linked genes are, and the smaller the map distance, the closer together the genes are on the chromosome, and the more likely those genes will be inherited together. Sex-linked traits are traits determined by genes located on sex chromosomes, and sex-linked traits differ from Mendelian traits because each parent does not contribute sex-linked alleles to every biological offspring, like with Mendelian inheritance. Biological sex will determine which sex-linked alleles are inherited. All right, that's it for AP Daily today, and I will see you next time. Welcome to AP Daily. This is Rachel Lytle from Brentwood, Tennessee at Brentwood High School, here to talk to you about topic 5.4, non-Mendelian genetics. So let's get started. What will we learn? We will learn how inheritance determined by multiple genes differs from Mendelian inheritance, and how non-nuclear inheritance differs from Mendelian inheritance. So there are many traits that are the product of multiple genes and/or physiological processes happening in combination. Many traits are the product of multiple genes acting in various combinations, and these traits do not segregate in Mendelian patterns. So let's look at an example of that. An example could be human hair color, which is determined by multiple genes. Alleles inherited from each parent for each gene have an additive effect in determining what the phenotype will be. So a dominant allele codes for dark pigment, and a recessive allele does not. The figure below shows possible genotypes and phenotypes if hair color was coded for by, let's say, genes A, B, and C. So you can see the possible genotypes here. If you are going to have a phenotype of the darkest hair color, that means you would have all 3 genes with all of the alleles as dominant alleles coding for dark pigment. So that's the darkest hair color possible. On the opposite end, you can see if you had all of the recessive alleles for all 3 genes, that would result in a phenotype of the lightest hair color because the recessive allele does not code for dark pigment. And somewhere in the middle, you could have any combination to have medium hair color of 3 dominant alleles and 3 recessive alleles. And notice that there's lots and lots of possibilities of genotypes that you could have there that would give you medium hair color. And so with traits that are determined by multiple genes, we often see a larger range of phenotypes, and most often there can be more possibilities of you having a phenotype somewhere in the middle just because of the way that the possibilities of genotypes line up like you see here. Right, some traits can result from what we call non-nuclear inheritance. So chloroplast and mitochondria, remember, contain their own non-nuclear genome. They have their own DNA. And chloroplasts and mitochondria are randomly assorted into gametes and daughter cells during cell division. The mitochondria are transmitted to the egg and not the sperm in animals. So that means that the mitochondria and their traits on their DNA are maternally inherited from the mother. Mitochondria and chloroplast are transmitted in the ovule and not in the pollen of plants. So those traits in plants for mitochondria and chloroplast in their DNA and the traits they carry are also maternally inherited. Traits determined by chloroplast and mitochondrial DNA do not follow simple Mendelian rules. So non-nuclear inheritance patterns then can be determined through pedigree analysis. So let's look at an example of that. Say we're looking at animals, and we know that the mitochondria are transmitted by the egg and not by the sperm. So the mitochondrial DNA that codes for certain traits are going to be maternally inherited. So if we look at that on a pedigree, it could look something like this. And you'll notice that each biological female, if she is affected and has the affected trait that's carried on the mitochondrial DNA, it will be transmitted to all of her offspring. So we see here that this this biological female is affected, and so she has transmitted the trait to her offspring. Again, an affected biological female transmits it to all of her offspring. And here we can see that we have an affected biological male, but that will not be transmitted to his offspring, because remember, the mitochondria are packaged in the egg and not the sperm. So he would not transmit that DNA onto his offspring. So we see this pattern of maternal inheritance here. All right, so what should we take away? Our key takeaways from this video are that inheritance is determined by multiple genes, has a large number of possible genotypes, and a larger range of phenotypes possible compared to the inheritance determined by a single gene. And these traits do not segregate in Mendelian patterns. Non-nuclear inheritance can be seen in chloroplast and mitochondria, and it is maternally inherited. And these traits also do not segregate in Mendelian patterns. All right, that's it for AP Daily, and I enjoyed talking to you, and I will see you later.
All right, welcome everybody. This is AP Biology, daily video for Unit 5, Topic 5.5, Environmental Effects on Phenotype. My name is Lisa Niesman. I'm coming to you from Bay Ridge Preparatory School in Brooklyn, New York. This is daily video number 1. This video will explain how environmental factors influence gene expression. The skill we're going to be focusing on is explaining biological processes in applied contexts. So let's get to it. What will we learn? In this video, we will learn what is phenotypic plasticity. We will learn how the same genotype can result in multiple phenotypes. And lastly, we will learn what effect environmental conditions have on gene expression. So our main point here is that the same genotype can result in multiple different phenotypes. And environmental factors can influence this gene expression, which means that if the environmental conditions change, expression of the gene can change. The fancy term that we use, we have phenotypic plasticity. This is the ability of one genotype to produce more than one phenotype. And this phenotypic diversity can be due to environmental factors, not necessarily due to genetic diversity, which means that organisms can have the same genes, but they can show different forms based on external factors. So let's look at an example of this. Examples always help clarify. The first example I want to show is flower color being based on soil pH. So I don't know if any of you have experience with hydrangea plants. There's a picture of them there in case you haven't seen hydrangeas. But what's really cool about them is that the color of the flower is actually determined by the pH of the soil. So what that means is that the same genes are yielding a different color flower simply depending on environmental conditions, in this case, pH of the soil, during flower development. In this particular case, an acidic soil, which is a low pH, is going to give us blue or lavender flowers, and a basic soil, which is a high pH, is going to give us pink or red flowers. I'm going to give you another example because examples are fun. The second example I want to look at is phenotypic plasticity in stomates. So in many different plant species, the number of stomates is actually determined by the amount of CO2 in the atmosphere. Again, this means that the same genes can yield something different, in this case, different numbers of stomates, depending on atmospheric conditions. Low amounts of CO2 lead to more stomates, and high amounts of CO2 lead to fewer stomates. Same genes, different phenotype. So let's practice this. What I have is a question that we're going to look at using this skill. So the skill we're going to be focusing on is concept explanation. And what that means is we're going to be asked to explain biological concepts, processes, and/or models in applied contexts. So I have a question here. This is a released AP exam question. And what I'd like you to do is to take a moment, read the question, think of what the best answer is going to be, mark that down somewhere, and then come back to me. So pause the video, try to answer this question, and then I will explain it for you. All right, welcome back. I hope that you paused it. I hope that you have read about mustard plant seedlings and that we're ready to tackle this question. So the question stem says, when a mustard plant seedling is transferred to an environment with higher carbon dioxide levels, the new leaves have a lower stomata to surface area ratio than do the seedling's original leaves. Which of the following best explains how the leaves from the same plant can have different stomatal densities when exposed to an elevated carbon dioxide level? So my hope is that as you were looking through this, you loved answer choice B, which says that leaf growth is promoted through increased photosynthesis, but the genetically regulated rate of stomatal production is not altered, which leads to a decrease in stomatal density. The key there is the genes are not changing, it's the environment that's changing, which causes that phenotypic plasticity. So we have a couple of big takeaways from this video. If you're taking notes, these are the things that I would make sure to write down. So our first is that phenotypic plasticity is the ability of one genotype to produce more than one phenotype. The same genotype can result in multiple phenotypes due to changes in environmental conditions. That was the changing pH or the changing CO2 levels in the atmosphere. And finally, environmental factors can influence how genes are expressed. So what we are actually seeing that organism represent. This leads to phenotypic plasticity. Thank you very much for watching this video. I hope this helped clarify topic 5.5 for you. If you have any questions, feel free to go back and watch the video. Good luck.